New research is always being done on cancer because there are so many areas to explore. But how do we know what's working? Many scientists who are studying cancer dive down into the cell's genes, looking at the DNA and RNA that are affecting the functions. But then they need to zoom out and look for more evidence of what these resulting functions are, such as how has the cell changed, and what's going on around the cell. The tumor cells and their surrounding elements are collectively called the Tumor Microenvironment, or TME. The TME is being explored in many studies for potential cancer treatments because researchers are realizing the importance of these extracellular components and their relationship to the tumor. The non-invasive microtest technology, or NMT, has recently been gaining popularity in the medical sciences and is contributing to a growing interest in studying the TME for potential cancer treatments. In this video, we're going to compare two similar cancer studies, one that used NMT and the other that didn't, to demonstrate how this technology and the tumor microenvironment can be used in cancer studies. So let's look at the first study that didn't use NMT and how they might benefit from the evidence it provides. This paper is about the effects of photodynamic therapy on cancer cells, looking at the responses and the cell death. These scientists worked with the expression and inhibition of genes and proteins for their study. Then they needed to look at the bigger picture to see the resulting functions. So they looked for these changes by observing cell morphology under a microscope and with an immunofluorescence test. These tests provide them with some information, but if they looked at the TME, they would gain more evidence of the functions. Perhaps the gene has caused more extracellular vesicles to be released. The tumor may have developed immune resistance. Maybe there is a huge efflux of calcium ions. This is where NMT comes in. The flux of ions and molecules in and out of cells is a very important part of the TME, as it provides excellent evidence for the functions of cells. Fluxes are the direct result of reactions in life processes. For example, if you were measuring oxygen flux in plant cells, you might see a large efflux of oxygen at one time. This would indicate to you that the plant is photosynthesizing, as oxygen is a product of that reaction. N.M.T measures these fluxes non-invasively and can test not only cells, but at the tissue and even organ level, providing flux data at many scales. N.M.T has a lot of appeal next to other tests as well. For example, remember this immunofluorescence picture? This and many other tests require labeling, something that is not necessary with N.M.T. So how can N.M.T be used in research? Let's look at the next paper. In this study, researchers investigated the effects of photodynamic therapy on cancer cells, looking at the cell responses and cell death, just like our previous study. But they also looked at the associated fluxes of calcium ions and oxygen. They found quick changes in the net fluxes after photodynamic therapy treatment. These measurements helped them discover that PDT was leading to cell death by generation of reactive oxygen species in the cells. They also realized that the fluxes they measured could be a monitoring system for future studies to use as indicators of cell death. Other treatments, such as drugs, may also cause cell death via reactive oxygen species generation. So, a future study could measure these calcium ion and oxygen fluxes in cancer cells, add their treatment, and then NMT's real-time measurement system would allow them to quickly see when cell death occurs based on these flux changes. But this is just one example of many cancer studies that could be done using NMT. Since the tumor microenvironment holds so much unexplored data on cancer, we want to do our part to help researchers investigate it by measuring the fluxes of ions and molecules. Every day, someone could be discovering a new gene that will change the course of cancer treatment, but do they have enough evidence for it? The fluxes of ions and molecules will reveal the truth, and we are here to reveal the functions in life sciences. So thanks for watching.